morning, everybody, and welcome to the Lungitude Foundation's Lung Transplant Research presentation for 2025. We're really excited about today. We've got some amazing speakers to talk with you, as well as a bit of a surprise announcement towards the end of the session, so stay on for that. My name is Wendy Jenkins, for those of you who don't know me, and I helped co-found the Lungitude Foundation, and we have an amazing board and fundraising committee and other teams joining us as well, which is how we're able to fund a lot of this research with your support. So we're very excited to be able to just share, where your money goes and what sort of things the team have been working on. So I would just like to start by acknowledging the traditional owners of the land on which we meet, and given we're in Melbourne, we're based here, that's the Wurundjeri people of the Kulin Nation. And I'd also like to pay my respects to any elders past, present and emerging, or who are joining us today. That's actually an image of the Yarra River, or my interpretation of, so I thought that was an appropriate one for today's session, seeing we're in Melbourne. So without further ado, I'll just quickly go through the agenda. So shortly I'll hand over to Associate Professor Bronwyn Levy, who has been involved with the Lungitude Foundation since the beginning, and she'll then introduce all the speakers that we're having, because she knows them so well, and she'll be able to talk to what they're doing as well. So these are the presenters for today. We've been a bit cheeky with the doctor there for Steven Hiho, because he has pretty much had his PhD approved. It's just formalities now, so we're going to call you doctor anyway, if that's all right with you. And then we'll have a panel Q&A towards the end, so that's when you can ask all your questions. So please, use the Q&A option to do that towards the end, or we'll allow people to type in questions then. I thought, just to give a bit of an overview of what we actually do at the Lungitude Foundation, I'd play one of our recent videos, which features Claire and Lee, who Claire is a double lung transplant recipient. She had cystic fibrosis, and her husband Lee is a great supporter, and they were actually the faces of our Giving Day campaign this year. So I'm just going to hope the technology works. I'm just going to play this for you. And then we'll move into the presentations.
I'm Claire. I grew up in the country in good old rural Victoria. For the majority of my life, I led a pretty normal life. In my late 20s, my health decided to decline. Transplant for my entire life was always a possibility. And then when I turned about 30, that's when the conversations with the doctors started. My lung function deteriorated to the point of needing a double lung transplant.
Hi, my name's Lee, and this is my lovely wife, Claire.
Lee and I met 16 years ago in a pub the old-fashioned way.
Got to know each other over the next week or so, and it was then that Claire told me that she had cystic fibrosis. The longer I was with Claire, seeing somebody that you're developing feelings for get to a point where lung transplant becomes a reality, it was really hard to watch.
I think the hardest thing I heard from the doctors pre-transplant was that if I didn't have a transplant, I would have 2 years to live, maybe. I got to the point where Lee and I lived in a double-storey house, and I would bring everything I needed downstairs because I couldn't walk up and down the stairs. I would basically just go from the couch to the bathroom to the couch, and that was about it. I think I used to use the term that I was surviving, not living, until my transplant. I remember in ICU, after having my transplant, I laughed hysterically at something without coughing. And that was something I'd never be able to do pre-transplant, which was pretty cool. Last year when Lee and I were thinking about doing a fundraiser, we did some research and found Lungitude, and really liked the way that they support research, the way that they're trying to make lung transplant patients' lives longer and easier, and the support that they give to transplant patients and their families.
Our key goal at the Lungitude Foundation is an important one and also a powerful one, which is transplant for life. We really want to focus on longevity for patients, and that's what a lot of our research and support programs, advocacy and education is focused on, to make a real impact in patients' lives.
At Lungitude, we don't receive any funding from government at all, so we really rely on you to fund a wide variety of research from the science lab to the bed that will help people not only hopefully live longer, but they can actually make an impact in the community as well.
I would say to someone that was waiting for a transplant or considering transplant that life is better after transplant, and you just have to get through the tough time and then you thrive.
Claire having a transplant's made a huge difference in the way that, our lives are led.
We think about the future, we plan to travel overseas, we plan to hopefully have a family somehow.
Your normal things that people do when they get married and grow old together, we want to do those things too, and a transplant has allowed us to have that opportunity.
Getting a puppy.
That's on the list, isn't it? Yeah. It's been really great to be involved with Lungitude.
If you want to support, it's quite easy just to jump on board.
Thank you so much for your support.
Yeah, thank you.
And we thank Claire and Lee for all their help with the videoing and their support this year. So great story. And I still don't know if they've got the puppy yet, but I have to keep an eye on that. So these are some of the other supporters who've been instrumental with helping us as well. And it's the foundation cannot exist without all of the supporters, and it doesn't matter how much people donate, it all makes such a big difference. So thank you to everyone. I'm now going to introduce Bronwyn, and just introduce with the slide there. Hopefully you can read all the wonderful things which she has done. She's been involved with clinical nursing for more than 40 years and has been involved outside the Alfred too, in a wider capacity with many different things, including Winston Churchill Fellowship and being part of many of the transplant and nursing organisations that you can see there. So a very vital cog in the wheel of lung transplantation. So over to you, Bronwyn, and I'll get you to introduce the first of our speakers.
Thanks very much, Wendy, and welcome everyone. And I'm going to introduce our first speaker that probably needs not a lot of introduction to many of you, Professor Greg Snell, who is our medical head of our lung transplant service here at the Alfred, and has been for over 30 years. He has a worldwide recognition for his contribution to really pioneering a whole lot of aspects of lung transplantation, and is a real leader both here in Australia and internationally. And he's going to give us a little overview of research, his ideas of research, where we're heading. He's just returned from a sabbatical overseas. He's come back with some great ideas, and also he can tell you about how well we're sitting within the international perspective of lung transplants. So without further ado, I'm going to pass over to Greg so he can start off proceedings today with his talk. Thank you.
Thank you for the introduction, Wendy and Bron. And I'm going to essentially just give a bit of a background of how we think about research. And I was struck as I heard Wendy give the sort of the story there of Lungitude, that really is our thinking too. We're working in parallel. The targets we have for lung transplant research is sort of multifactorial. The first thing we think of is getting as many lungs into as many people as possible. And the way we do that is to work in the background, looking at the organ donation system, educating them as to what we can use, what we think we can use, what can they bring towards us. We've more recently looked at new types of lung donors, and I'll talk a little bit about that with just a couple of pictures in coming slides. But using not just the traditional sources of donors, but trying to use different parts of the donor pool of people who are prepared to donate their organs through the different ways that you can be in hospital, in an ICU with brain problems or whole body problems, or indeed nowadays where you can have a voluntary assisted dying pathway and donate organs on the back of that. We've looked at organs that are not perfect, and there's obvious things of using older organs, and those are from people who've smoked, and we've got really a pretty good idea where the boundaries lie. The other thing we've been doing in more recent times is talking about changing the logistics. We've traditionally used an esky to bring organs back, but it now seems that you can be a bit fancier and use a refrigerator, and I'll talk about that. And the idea of also improving the logistics so that the life balance is a bit better for the at least for the surgeons, or the rest of us might still be up in the middle of the night. And in terms of looking at organs that we wouldn't have traditionally used, this is a couple of pictures from my sabbatical. This is a tiny village on the left in the middle of nowhere with what used to be a phone box, which now is a defibrillator store. And that's in Lower Slaughter, I think, actually, in the middle of nowhere in the UK. And the next one's in downtown London, just street defibrillators. And the truth is that that can save your life, but if you don't quite get there, and you don't survive your cardiac arrest, then the lungs can actually be used, and that's the type of thing we're working towards, saying, well, there's the number of people who die in hospital is very small. The number of people who die sort of out in the community who end up with a sort of a final pathway through the hospitals is much bigger. The pictures here show a retrieval area in Papworth, where they're currently getting, to the pictures on the wall, kidneys and livers from people who collapse out of hospital, and they've got a specialist sort of centre there that they use those organs. And I was there saying, you need to get lungs. Lungs are easier to get, and was actually able to put the people down the road who do the lung transplants in contact with the people who are doing the out-of-hospital retrieval of kidneys and livers. And that's the type of thing we want to head towards here. There's a lot of organs out there that could be used that way. The second point on this front slide was the eskies and now using a fridge. You might say that fridge looks more like Gordon's wine fridge, but it's the truth is, it's turned out that you maintain a higher temperature and you can get organs that are 13 and 14 hours after someone has died. And you have a situation then where you can basically avoid the nighttime operating, which has been a problem for the teams. And this is not exactly an Alfred operating room in the middle of the night, but it's clearly 2 o'clock in the morning is not the best time for surgeons and anaesthetists to be doing their best work. So we're working towards this one. The other targets for research, we want to improve the early outcomes, protecting the lungs by clever ways to stop them being damaged through the transplant process, protecting the kidneys so they're not damaged, reducing the CMV infections that are very common after transplant, trying to understand also what can make lungs work better in the long term. Why don't they all come out at 100% a year afterwards? What's this rejection that we talk about? How do we diagnose it? How do we prevent it? What clever steps can we make with the matching? And if we've got any clever steps to treat things, and improving, general health, looking at osteoporosis, skin cancers, and some of these other things. They're important to the people who've had a transplant. They're very important to us. We want our work to be easier and better as well. How do we get ideas? Well, how do we get transplant research happening? Well, basically, we think of things that are not quite right and try and fix them. We hear of things from overseas that we say, actually, we do that better than you're doing it, so we'll tell you. And we get people come to us with ideas of novel equipment or novel medicines, and they come to us and say, can you see if this works? The team is anyone who wants to work with us, really. There's a core team from of the Alfred staff, but we have lots of people we work with, and obviously some on the panel here from locally, interstate, and overseas. The practical support, administration, planning, etc., clinical support comes from Bronwyn leading the team, and No and Lucy in the background doing the sort of running around with ethics and working with the patients. We've had local and overseas fellows, and that's looking like being very successful going forward. We've had I think, 4 or 5 applications of overseas people to come and work with us for protracted periods. And then we've got the laboratory and sort of team in the background who are working on some of the sort of more laboratory and scientific based bits and pieces. Lucy, Steve, Brad, Julia, and Bridget. How do we get funding to resource this? Well, we work harder and longer, and we do this, from our sort of salary base here at the Alfred. We seek grants from the government, and you'll hear more about that in time. We seek grants from Lungitude. We get some specific donations via Lungitude from individuals, or sometimes from the Alfred Foundation. We go out and chase trials and equipment. We don't get specific funds we don't get non-specific funds from the Alfred at all. The Alfred Foundation doesn't raise money for lung transplant themselves, and we're trying to work on that. And the university does not raise any funds for lung transplantation. So it really does come down to the individual work that we've talked about, Wendy's talking about. How do we translate this back? Well, basically, abstracts, presentations, talks, visits. We publish scientific papers, we change what we do, and then we look to see whether that improves results, and we balance that against the national and international registries. We jump up at meetings and grab the microphone and tell people what they're doing wrong, or ask tough questions. We talk to the government, and we, as I mentioned, try and talk to some of the junior people all around the world, some who we can bring here, some of whom we can work with and turn out to be great scientists in their own right in other places overseas. My sabbatical was about that in some ways, scouring the world for ideas and contributions, making transplant great again. And of course, there's experts out there who've made whole careers out of improving, not improving, probably not the word, and probably enhancing everything around them to push their own interests. And so I've been to the source of some of these things to try and improve things. In conclusion, I think our lung transplant research aims to improve outcomes for everyone, patients, the hospital, the taxpayer, and the team here. And we aim to translate them, not just to talk about what someone else should do. And we aim to tell the world. And I have to thank everyone on this presentation for their generous support and its moral advocacy, involvement in studies, by patients, and thank you to everyone. So thank you very much.
Thanks so much, Greg, for that great introduction. And I just wanted to remind everyone to, if you have any questions, there's a Q&A button down the bottom of the screen there. Put your questions, you can type your questions into that, not into the chat button, but the Q&A button there. And then hopefully we'll have time to respond to those towards the end. So the next speaker I'm going to introduce today is Dr. Miranda Paraskeva, and Miranda has been working with our lung transplant program for quite some time as well, and is now head of the Paediatric Lung Transplant Service here. And she is pretty active in the research space as well, particularly working with the transition of paediatric patients to adult care, as well as managing the paediatric transplant program that's the Alfred Hospital is the home for. And without further ado, I'm going to hand over to Miranda now for her to do her presentation.
Thank you for giving me the opportunity to talk. So our paediatric group was the recipient of the inaugural Zoe Brooks Paediatric Grant. And so our research is a little bit of a different focus to some of the other things you'll hear about. Essentially, what we're planning to do is, or in the process of doing, is co-designing an animated education resource for paediatric lung transplant recipients. So I'm sure you all know, but I know that the majority of people in the audience are adults, is that lung transplantation also occurs for children. And whilst being the largest adult lung transplant centre, we also host the nationally funded Centre for Paediatric Heart, Lung and Lung Transplantation. So we're the only provider of lung transplants for children aged 4 to 16 in Australia. So when we look at sort of the last 19 years, we've done 65 transplants in 64 recipients, predominantly bilateral lung transplants with a few heart lungs. interestingly more in girls than boys, with an age range of 4 to 17 and about a median age of 13. And we've also assessed a number of kids that have aged out of the paediatric program whilst waiting for their transplant. But Australia's a big place, and children from all over Australia travel to Melbourne for their transplant. They spend the next 3 months for rehabilitation prior to returning to their home state for ongoing care. And what that means is that this centralisation of surgical and medical expertise needs us to be able to provide education to these children and their families at a distance with the support of their local healthcare providers. Okay. So paediatric education is a little bit different to adults. It needs to be tailored. Childhood and adolescence is a time of complex and rapid change, and kids' cognitive skills are also shaped by how healthy they are, whether they've had opportunities to learn, how much school they've missed. But in the context of lung transplant, that's also impacted by hypoxia, spending a lot of time in hospital, fatigue, and disrupted school. And so we've looked at at how we can improve education so it's developmentally appropriate in both the delivery and the content. So we asked children and families what they wanted, and this was a long time ago. It's taken a long time to get funding for this project. And basically we did a survey and asked children, their families and their healthcare providers what would be useful. And basically what they said was that there was a lack of age-appropriate, child-friendly resources that taught them about basic anatomy, about their condition, about transplant, about the transplant procedure, how they recover, and how to live well after transplant. And what they also told us was that they would prefer things to be web-based and animation rather than real-life pictures. And the healthcare providers told us very similar things. And so what we're aiming to do is design an education resource for children that explains transplant in a way that they can understand, but also meets their developmental needs. And what we're planning to do is engage children and their carers in the process to co-design the animation. So our team, including myself, is members of the Paediatric MDT, Simone West, who's our OT, and Jane Miller, who's our psychologist, as well as Jess Kurz, who is an animator who works for Monash as part of the School of Design. And then we have a project advisory group of clinicians who've worked in transplantation, those who've referred children for transplant, recipients who've been transplanted as children and now as adults, and carers of children who have received a transplant. And what the process is there's been a development of a script by our team that's been reviewed by the project advisory group for content accuracy and medical suitability. And then we'll be recruiting 2 groups, children who've had a transplant and caregivers who've had children who've had a transplant. And whilst the animator will design the initial storyboarding of the animation, there'll be 2 sessions where these 2 groups of recipients and their carers will be involved in shaping what the animation looks like and how it sounds. So the script has been written, and these are sort of excerpts of the script. And basically what it looks at is this animation will describe the initial point of transplant from the surgery to discharge. We'll then be recruiting children with a transplant and their carers and undertaking the co-design sessions. So Jess and Simone will run 2 sessions. The first session will focus on the content and visual design, whether the group finds it relevant and acceptable, whether they like the colours, the movements, the animations. And then the animation will be finalised. At that point, the second session will look at the animation, the voiceover and the music with a plan that will be completed based on the feedback. And where we're up to at the moment is Jess working on the animation and storyboarding design, having had feedback from the project advisory group. And this is just an example of Jess's work. So animations take a little bit of time to prepare, and so we're now at the mercy of how long it takes to draw the pictures, and we'll keep you updated with how we progress.
Fantastic. Thanks very much, Miranda, and we'll really look forward to seeing this project as it evolves and hopefully see the actual whole animation in the next year or so. So we're going to change tack here a little bit now to away from the clinical side of things to hearing from Dr. Steven Hiho, who is from the tissue typing lab in Victoria. It's called Victorian Transplant Immunogenetics Service, VITAS as we call it. Steve is an immunologist who's just finished his PhD and has been working with our team for many years. His PhD's probably taken about 5 years all up, and he was awarded a Lungitude Foundation grant in the last 12 months, and he's going to do his presentation for you now on this project, this specific project. So I will hand over to you, Steve.
Well, thank you for the introduction, and thank you for everyone joining us and giving me the opportunity to talk through the project that was supported by Lungitude. So the support was for looking into transfusions in lung transplantation, and particularly the HLA sensitization and impact on outcomes. So what is the need for blood products? Well, lung transplant, as we know, is major surgery. There's complexity involved. It often leads to a lot of need for blood products, particularly red blood cells, which increase the oxygen through bleeding, for platelets to help clotting, and also for plasma. All these are very important to stabilize after transplant and in a very short period during the transplantation surgery itself, and around 70% of lung transplant recipients require at least one transfusion. So what is the risk of these transfusions? Well, once you get a transplant, as I said, you often need transfusions, but with this becomes the risk of creating new antibodies. So these antibodies can potentially provide an opportunity for the body to sort of reject that transplanted organ. It can lead to worse transplant outcomes, or in the need of a subsequent transplant, it could reduce the chance of finding another donor. So these antibodies that we talk of, I've spoken to this group a couple of times about some previous projects about HLA, or human leukocyte antigens. So HLA is a cell surface protein. It's responsible for regulating immune responses. So HLA is important in determining whether a foreign body is there. It can look at the HLA on that foreign body and work out that if it's non-self or if it's self. If it's self, there's no immune response. But if it recognizes it as non-self, it can lead to a cellular or T cell response or a humoral B cell antibody response. So why is this important in transfusion? So HLA expressed on a multitude of cells. It is highly expressed on platelets with high class 1 HLA A and B expression. And although it's not expressed on red blood cells, there are residual white blood cells within the blood cell donation, and those white cells do express both class 1 and class 2. And in plasma, there's potential for soluble HLA, which all can lead to an increased risk of producing antibodies against these HLA targets. So what do we know about transfusion in transplants? Well, this is an Australian study out of South Australia. It looked at kidney transplantation. They looked at the development of antibodies, and they didn't show a significant difference in the outcomes for those that were transfused versus those that were not transfused. However, this is another kidney study of about 75 kidney recipients looking at antibodies developed directly from the transfused product themselves. So in total, 57 recipients received almost 200 red blood cells. So they only looked at red cell transfusions. And of those, 50 of them had at least one donor typing. What they did find were antibodies that were developed to the transfused product, most notably the HLA-B of class 1, but also there was the class 2 DQ as well. They did state that these transfused specific antibodies were transient, so they were short-lived. However, there's a potential for them to be able to create a memory response later down the track. So in lungs, there's not a lot written or published in the lungs about transfusion products. This is one out of Houston where 232 lung transplant recipients, 68% received at least one transfusion. They looked at outcomes only within the first 30 days. They did not look at any of the HLA mismatching or antibodies, but they did find a poorer outcome to those that had transfusions. So what is the impact of HLA mismatching on a red blood cell? This is a recent project that was performed at Lifeblood. It looked at Victorian adult and actually pediatric kidney transplant recipients. They had multiple arms, but More notably, there was a HLA compatible or a selected arm where only selected red cell products were used, and then a random unselected red blood cell products were used. They looked at post-transfusion antibody screening, and as you can see there, those that were selected for lower HLA mismatching or better matched had less antibodies. There were some that had some class 2 antibodies within the project, but this was later found to be a pre-sensitization event. So what did that lead to? So that led to the lifeblood being able to supply compatible red cells. So these are particularly for kidney patients, mainly almost majority pediatric patients that are awaiting a transplant. We can provide them, or we now provide them, the lowest HLA mismatched red blood cells. So this reduces the risk of antibody formation while they're waiting for either a living-related transplant or if they're on the donor list. So they're keeping their donor pool large without those antibodies. So what have we found at Alfred in our study? So we looked at 300 recipients who received at least one transfusion. Now this transfusion was either red blood cell, plasma, or platelet. So looking at the patient's HLA and HLA antibody status, but we also have the database of over 40,000 typed HLA donors at Lifeblood to draw in on. And our aim was to look at the impact of these HLA in the transfusion on the outcomes and sensitization. So what have we found? So probably not surprising, we found that the more transfusion equaled poorer outcomes. Now, this is a very complex thing. Obviously, those requiring more transfusions have a lot more going on than those that required least. This was the same if we individualed for red cells, plasma, and platelets. So were there increased antibodies? Well, there was a trend towards increased antibodies for those that had more transfusions. However, this was not a significant difference between those. And once again, that makes more sense. As I said, there's more exposure to more risk of HLA antibody formation through the greater use of blood products. However, the HLA mismatching between those for those transfusions, while there was a slight increase in antibody production for those with high mismatching, there was no significant difference. So this was just for the HLA class 1 that we looked at the moment. We currently have to tease through the HLA class 2 and its complexity to work out if that forms the same trend. So where do these antibodies come from? Well, hopefully you can see there on the left-hand side is the increased HLA mismatching. So the one toward the lower towards the x-axis is better matched and the higher is worse matched. Those with no antibodies, there was no difference between those or those that had antibodies to the transplanted organ or those that had antibodies to the transfused antibody from the transfusion. And once again, this didn't show any association with graft outcome either for those with transfusion antibodies versus those with transplant antibodies. So in summary, the increased number of transfusions were associated with poorer graft survival. And as I said, that's probably not unexpected. The increased number of transfusions did not associate with an increased antibody development, particularly HLA, and that was HLA class 1. HLA mismatching of transfusions did not associate with poorer graft survival or increased antibodies. However, there were evidence of transient transfusion-specific antibodies, and this probably needs to be looked at a little bit further. And we aim to look at if certain mismatches need to be avoided in these transfused products. So what's ahead? So we've still got a little bit of gaps to fill in the donor typing and getting more units. So more units on this database. We're going to tease out the complexity of each of the product types, the red cells, the platelets, and the plasma. And we haven't really dug into the class 2 level of mismatching yet for this, but we aim to do that very shortly. And are platelet and red cell mismatches the same? If so, what can we do? And with the HLA mismatches, is there a cumulative effect? If the mismatch is the same for the transfusion product as it is for the transplant itself, does that become worse off? So these are things that we have to look for. And once again, I'd like to thank everyone at Lifeblood and the Alfred and obviously the Lungitude Foundation for their continued support through the years and projects. And hopefully that was all okay.
Fantastic work, Steve. It's so complex. I think it's amazing how you can even tease out all of that in so many patients with so many transfusions. So we look forward to hearing a bit more about that as you continue the work. Okay, and moving on to our next expert from the research labs and from immunology as well, Dr. Nicole Mifsud. She's a group leader in clinical immunology in the Department of Biochem, Molecular Biology and Biomedicine Discovery Institute at Monash. And she is an expert in T-cell immunobiology and HLA immunogenetics as well. And over the years, Nicole has done various projects that have been supported by Lungitude too. And so we're going to ask her to present this latest project that she's had funded in the last 12 months. So I'm going to hand over to you, Nicole.
Thanks again, Bronwyn. It's a pleasure again to be here to talk about the projects that are really funded by the Lungitude Foundation. So we've been focusing on a project that's really trying to distinguish between T-cell heroes and villains in order to improve transplant outcomes. So every day we have immune cells that circulate throughout the body, and some of those immune cells are referred to as T cells. And what T cells do is they really try and survey the landscape to see if there's any foreign entities. And so Steve sort of introduced this earlier, where he's talked about, looking at self in the setting of both the HLA and also in the peptides that they produce. So in the absence of any sort of foreign entity, these T cells remain inactive, and so there's no immune response. However, this landscape can change quite considerably when you have a viral infection. We now have the virus that's infecting these cells, and the cells themselves can actually present parts of that virus in terms of peptides to the T cells that are circulating. These T cells become activated, and they can actually kill these virus-infected cells. And this is a very important part of what we call protective immunity. In the setting of transplantation, the recipients also have a very foreign landscape when they receive the donor organs. And so you can see here on the donor cells, we have different HLA peptide complexes that are presented to the different circulating T cells. These T cells then become activated, and they can induce very strong immune responses that can actually cause graft rejection. And these T cells are referred to as alloreactive T cells. So this project was really trying to differentiate between the heroes that we need for everyday protection and the villains that are really contributing to graft rejection. So how we went about this was really to try and generate some alloreactive T cells. So these are the T cells that see different HLA mismatches, to then try and characterize the receptors that they're presenting, so what their different signatures are, and then also to examine the features of those different T cell receptors and how they're associated with anti-graft immune responses. And so to do this, we really concentrated on a very high frequency HLA allele called HLA-A2, but we looked at it from a haplotype perspective in order to try and capture some of the other HLA. So what a HLA haplotype means is that we have a series of genes that are inherited as a single block. So the way we go about this is to perform what we call as a mixed lymphocyte reaction, which is basically a transplant in the laboratory. So we take some blood cells from one individual, we term them the recipient. We take some blood cells from another individual, we term them the donor, and we look in this direction and try and generate T cell responses against the mismatches that are represented on the donor cells. This happens over a period of time in culture, and as you can see here, we have pools of cells, each of those directed to the mismatch. We can then quantitate this in the laboratory, and we use instrumentation called flow cytometry to be able to determine the immune response to each of those mismatches. What's interesting, though, is that when we look at different recipients, so we have 3 different recipients here against the same donor, we'll see that the hierarchy as well as the different alleles that they respond to are different. And this really drives home the concept that it's not just about the donor HLA that's important, but the recipient's HLA is also important. And this really accounts for some of that variation. So when we performed the different mixed lymphocyte reactions across the 4 different haplotypes, you can see this is haplotype 1, 2, 3, and 4. So there's a total of 45 different mixed lymphocyte reactions. You'll notice that we can see variability in the immune responses to the different alleles. So in this case here, if we take haplotype 1, these are the 20 different recipients against one common donor cell. You can see that there's a huge number of variations in terms of the way that they can recognise the HLA-A2, the B7 and the C7. And again, as mentioned, this really capitulates that point that, you know, the recipient HLA is equally important, as is the donor HLA. What you'll see also boxed in red are the highest levels of immune reactivity that we saw for each of those haplotypes. And most of that was directed towards HLA-B, with HLA-A also showing some very strong immune responses, but very low levels of immune responses were seen to the different HLA-Cs. And this is not something that's uncommonly known. So this is looking at T cells as a pool, and then what we have now is some really wonderful technology where we can really dive deep into looking at each of these responses for the recipients at a single cell level. So we really wanted to focus on haplotype 1 and looking at responses to HLA-A2. So to do this, we have a workflow called single-cell transcriptomics, but basically what it means is that we took all those pools of T cells from a recipient-donor combination. We were then able to stimulate it with a cell line that expresses a particular HLA. In this case, it's HLA-A2, and we can really focus all our efforts on really dissecting out the immune response to those T cells that recognize A2. We then stain them. We use a sorter so that we can really look at the CD8-positive T cells that were directed against that A2 allele. We can then perform some laboratory-based techniques to sequence this information. So we have a lot of information that we're trawling through at the moment. So I just want to cherry pick just a few examples because it's really quite complicated analysis. So on the right-hand side here is really just a color chart, and it's really going from one single T cell or one T cell receptor all the way up to hyper-expanded T cell receptors where there's more than 500 clonotypes of that single T cell. So when we're looking at the responses to the A2, we're looking at this activated column here. This is the inactivated cells. And what you can notice is as you're going down this color chart is that you're decreasing the number of T cells that are clonally expanding. And this is important because we think they're the ones that are actually going to be contributing to any kind of immune response in the setting of a transplant rejection. So what you'll notice in these two individuals, as you increase the clonal expansion, you decrease the number of T cells that are involved. And this is really important because it's not like we need to look for hundreds of thousands of T cells to worry about in terms of which ones are causing the damage. It's quite clear that it's only a handful of T cells that are causing problems in the setting of a transplant scenario. So to take that even further, when we looked at this one individual, we're able to identify what the T cell receptor signature is, and we can do that through the analysis platforms that we have. But this one T cell represented almost 40% of the overall response in this mixed lymphocyte reaction. It's hyperexpanded. There was more than 700 clonotypes for this one T cell receptor. And so we wanted to ask, well, what transcripts or genes is it expressing, and are those genes what we would typically see in the setting of transplant rejection? And so when we look at this, we can see on the right-hand side, again, just looking at the interferon gamma positive, so these are the activated T cells that are directed against A2, we can see a lot of these green boxes. And what the green boxes refer to is a signature that's showing that the T cell is activated and it's capable of producing cytotoxic molecules and has an effector status that would be related to a graft rejection response. So as I mentioned earlier, looking through the different mixed lymphocyte reactions, we're building these personalized maps for each of the mixed lymphocyte reactions. And we're getting a sense of how many T cells are involved and also what their biomarkers are. So in summary, this project is being really a great insight in looking at the alloreactive T cells, not just to A2. We do have data on the other alleles, but we just haven't examined them at the single cell level as yet. But we look at the T cell signatures and we can look at the effector features that we would consider be important in transplant rejection. we show that really there's only a small number of hyperexpanded T cell receptors. And this really allows us to think about, how we can use this for immune monitoring post-transplant, or even the potential of developing some kind of therapeutic. And again, these hyperexpanded T cell receptors have this signature that's pro-inflammatory and cytotoxic. What I haven't been able to show you today, just because of the length of time for the presentation, was that most of these T cell receptors have a high degree of diversity between the different recipients. So there's not an overlapping repertoire between individuals. And this really leads to the fact that we should be moving more towards a personalized medicine approach and not just putting everybody in the same category. And again, that would lead to some better downstream post-transplant monitoring approaches. This just really leads me to thank everybody that's really involved in my research, but more importantly, I think for this project has been Hayley Tucker. So she's a PhD student of mine, and so this work is contributing to her thesis. Of course, the blood samples that were used for this project were from the Australian Red Cross Lifeblood, and of course, this work wouldn't really be able to be conducted without the generosity of the Lungitude Foundation. So thank you very much.
Fantastic work there, Nicole. Very complex, but that's right back down to the cellular level. So, and that's where we all start from, cells. Our last speaker for today is Dr. Melanie Wong, who's a respiratory advanced trainee within our respiratory department here at the Alfred Hospital. And Melanie has been working with our respiratory department now for a few years and undertook a project that was funded by Lungitude Foundation in the last 12 months. And I'm going to let her introduce her topic and pass over to her and her slides for the next presentation.
Thank you for letting me join this meeting. I'm Melanie, and I did a project with the lung transplant service at the Alfred looking at an overall 30-year experience of lung transplantation for pulmonary sarcoid. Just want to thank my co-authors listed there from helping with this project. So I don't have any conflicts to disclose. And again, thank you for the Lungitude Foundation for supporting this. So just a general background for my topic, pulmonary sarcoidosis. It's labeled as an uncontrolled immune response leading to non-necrotizing granulomatous formation. It typically affects quite young adults between the age of 25 to 40. It's often a self-limiting, very mild disease. However, there is a small proportion that progresses to end-stage fibrotic lung disease. And those that continue to progress despite immunosuppression are those that may require lung transplant. Issued estimated around 2.4% of lung transplant recipients were transplanted for pulmonary sarcoid between 1995 and 2018. And it is the most common disease to reoccur post-transplant, with studies varying between 14% and up to 47%. So in this project, we sought to evaluate characteristics and long-term outcomes of patients following lung transplant for pulmonary sarcoid, and looking at risk factors and the impact of disease recurrence following lung transplant. It was a retrospective observational cohort study, and we looked at all consecutive patients who underwent lung transplant for pulmonary sarcoid at the Alfred between January 1994 to March 2024. We looked at their demographics, spirometry, evidence of rejection and granulomatous disease on transbronchial biopsies, imaging results, as well as hospitalization and their immunosuppression. So our cohort was quite similar to worldwide data in terms of having 2.5% of lung transplants that were for pulmonary sarcoid in our overall cohort. And in the end, we had 40 patients that were involved in our data analysis. There was a mean age of 51 years, and there was a male predominance. 5% had concurrent cutaneous sarcoid, and 22% had concurrent cardiac sarcoid along with their pulmonary sarcoid disease. Looking at infections in these cohorts, the most common infections were viral infections, followed by Aspergillus and Pseudomonas, and there wasn't any particular difference between patients who had recurrent disease and patients who did not post-transplant. Looking at overall deaths at the time of medical record review, just over half of the patients that we looked at had died at this point. Pretty equivalent proportions in patients who had recurrent disease and those who did not. And the most common cause of death in these patients was infection, followed by chronic rejection. So looking further into those who had recurrence of pulmonary sarcoid following their transplant, this occurred in 35% of patients, or 14 patients, and it recurred at a median of 15 months post-transplant. Surprisingly, the cohort that had recurrence were younger at the time of transplant, those without recurrence. And one patient who did have disease recurrence required a redo lung transplant for it. There was no statistical difference that we found for spirometry results, immunosuppression, or the number of hospitalizations between these two groups. So we looked at how we diagnosed sarcoid recurrence, and it was pretty evenly proportioned between those who were diagnosed on CT, those diagnosed on transbronchial biopsy, and those that were diagnosed with both. Looking at CT findings, the most common CT finding that was associated with sarcoid recurrence was right upper lobe nodules. And this is in comparison to the most common CT finding pre-transplant for pulmonary sarcoid, which was upper lobe fibrosis. Looking at transbronchial biopsies, we looked at 220 transbronchial biopsies overall. 105 were from patients with recurrence. Of those with recurrence, around 14% had non-necrotizing granulomas on those transbronchial biopsies, and 9 of those 14 were outside of the routine post-transplant transbronchial biopsy system at a median of 9 months, mainly for investigation of a drop in lung function. 4 patients overall had acute rejection on their transbronchial biopsy. 2 of these were patients with sarcoid recurrence, and this was picked up within 2 months post-transplant, so quite early on. This is just an overall summary of the patients that we looked at and what they were managed with at the time of recurrence of pulmonary sarcoid. So 3 of them were treated with infliximab, which is a TNF-alpha inhibitor. 5 were treated with higher doses of prednisolone. 1 of them did not have any change to their immunosuppression or their medications. And the rest, there was no available data that we could find as to specific treatment for sarcoid recurrence. So looking at the 3 patients that received infliximab for sarcoid recurrence, so the red X's are FDG avid on PET. The yellow is some reduction in avidity on PET, and the black zeros are areas that are not avid, and we're looking at different lobes in the lung: right upper lobe, middle lobe, lower lobe, left side, and mediastinal lymphadenopathy. So 2 of them had complete metabolic response, 1 5 months and 1 15 months after receiving infliximab. The third one, it was a bit unclear, mainly because they also had a concurrent Aspergillus endocardiae infection at that time. So whether that avidity was due to infection or sarcoid was a bit unclear. Looking at overall survival between those with sarcoid recurrence and those without, there was no statistical difference in overall survival between these patients. However, it did appear that there was a trend towards slightly improved survival with sarcoid recurrent cohort. In terms of limitations of our study, it is a single center study. It was retrospective, and the cohort size was 40 patients, which is quite small. And so definitely larger prospective registry-based data is required to further investigate this. So overall, we found that sarcoid recurrence was common, very similar to rates in other studies. However, it did not significantly impact survival, lung function, or frequency of hospitalization. If we were to want to diagnose recurrence and actively look for it, we found that right upper lobe nodules on CT were more associated with it. The detection of disease recurrence in terms of non-granulomatous disease on a transbronchial was largely found at 9 months, and whether there should be part of surveillance and routine transbronchial biopsies for patients who are transplanted for sarcoid. And infliximab, which has been used and studied in pulmonary sarcoid outside of the transplant field, can be considered as steroid-sparing agent for sarcoid recurrence post-transplant. However, we definitely do need more studies and more evidence for this, as our study only had 3 patients that was treated with it, but it does have interesting and hopefully promising results. Those were my references, and that's my overall study. Thanks.
Thanks very much, Mel, for that interesting presentation. And there's a few claps going on there from the audience, which is great to see. So that actually concludes all our presenters' talks today. And we do have a little bit of time set aside for some Q&A. As we said before, if you'd like to ask a question, and this is Wendy's just put it up here, you use the Q&A option at the bottom of your menu, and you can just type a question in there. That's the easiest way, because there's too many people on to open up for everyone to speak. I'm just going to read out a comment from one of the attendees. Who said, these presentations just highlight how dedicated you all are, i.e. all the researchers, and I can be amazed at the great results you're able to achieve with so much more research needed, and how complex the complexities of transplantation really is. These presentations place transplantation results in real context, and she remains very grateful for the fantastic results you all deliver, and says thank you. And I would actually like to say that, I feel very privileged to work in such a great team, which has enabled so much great research to be done over many years. And I think without Lungitude's Foundation support, a lot of these projects wouldn't have been possible. So, it's really important to recognise that the funding that we get through organisations like Lungitude to help us look at how we can improve outcomes and make transplantation better. I'm just looking here now. Someone's asked, Greg's already typing away there, is sarcoid similar to IPF? Greg, would you like to actually answer that in person?
So the answer is yes and no. If you just look at a chest X-ray, you might think that they're the same. Pulmonary fibrosis and a lot of the scarring conditions of the lung are in the bottom of the lung, which is where all the blood goes with gravity. And so sarcoid's often tolerated a bit because it's in the top of the lung. Sarcoid's pretty weird because, as Melanie said, it sort of you can find some of the changes on the biopsies within weeks after a transplant. So it's like it's it almost begs the question whether it sort of floats around the body a little bit as a different type of cells, or whether it's even some sort of infection. And it does respond in the majority of people can avoid a transplant with sarcoid because it's actually, it does often respond to steroids or just burns itself out. So it's a rare condition, but it's quite, it's almost, each person gets their own individual pattern for it. So it's an interesting condition.
And there's a question from Miranda here. Are you designing a resource for teens, not just for pediatrics per se?
Yeah, so I think the focus of this animation, the age group we've sort of targeted at is sort of the 7 to 13, 14, but certainly it could be used by anyone. I think, are we currently designing resources for teens specifically? No here, but there's a lot of work going on sort of at an international level with ISHLT, who are working on transition resources, so resources for young or teens and young adults to manage transplantation and, the transition to independent care. And so I'm part of a working group that's working on that. And so hopefully we'll at some point also develop some local resources. But the current focus is on the paediatric group with this project.
I'm thinking actually that we could even do something better for adults. And I think animation's a great way to do it. But any good producers out there that are interested and helping us update our adult education. That would be fantastic. Nicole, I'm going to ask you a question. How, obviously, the immune system's really complex, and, you're unraveling sort of stuff at a really minute cellular level here. It's sometimes hard to see how long it takes for anything that you find in your research to actually get into, say, clinical practice. What are you thinking is a sort of like a timeline of and I know that's putting you on the spot, but I think it's, sometimes it's hard to sort of think, okay, and it's like a building block. But yeah, what's your thinking about what you're working on in that project?
Thanks for the question. It is a difficult one. It's one I get asked all the time, and we sort of throw a 5-year plan out there. But the reality is I guess, we're really at the start of looking at single cells in the setting of transplantation. We're trying to identify T cells that are really quite destructive, and seeing whether they're public or not. So they're in more than one individual. So at the moment, everything sort of leads that it's more personalised. Which would make, trying to generate a therapeutic a little bit more difficult. So we're really trying to look at public T cell receptors. It's taking time. We've got some new data in another project which indicates that can be achieved. But, what we're hoping for in the medium term is something about immune monitoring. So we'll be able to look at individual patients, monitor to them better, and then perhaps look down the road for a therapeutic. And I think that can be achieved within 5 years.
And when you say immune monitoring, because already we look at patients pre-transplant and take blood and look at like what their HLA is and their risk. So would you be thinking that you would do blood pre-transplant as well as post, and then because you've got the donor, impact of the donor as well, and so you've sort of got what their profile is pre and then what's happening post? Is that the sort of thing that?
Yeah, absolutely. So what we're looking for is something to sort of tack on to already the routine biopsies that are being done, but at a blood test level. So we're still trying to evaluate what we're seeing in the periphery in the blood is actually matching what is actually happening in the transplanted organ. And we are seeing some shared T cells again that are coming up. And so, yeah, we're hopefully having a blood-based test where we could look at the level of the T cells to see when they're starting to expand and give some information back to the clinicians about that process.
Great, thank you. Steve, I'm asking all the questions here because no one else is asking. So I'm going to ask you about transfusions. Obviously, the Red Cross Blood Bank, does all of the blood banking and matching of all the blood products that are distributed around Australia, but obviously matching every single unit of blood product that a patient gets after transplant it would be very time consuming and expensive. So, what's your thinking about the importance and the sort of impact and cost that might happen on the blood bank and the team at the immunology lab for that?
Oh, well, there would be a huge impact. What was recently done with the red cells in the kids' kidneys, as I suggested, that a lot of that is pre-transplant. So they've got a transplant time that they can work to and they can plan ahead, and it's only a handful of units. From what I've seen with the use of transfusion in lungs in the post setting, it's probably not ideal to get matching. It would almost be impossible. Even with 40,000 donors that we have typed, it would almost be impossible to get matched at some of what the patients require. So it's a matter of just teasing out, is there a need for it? Is there any mismatches in particular that we need to avoid? And that will just give us a bigger pool to use. But it's a bit of work to go into it. But yeah, it's a lot of work to do, especially when so many units are required so quickly in the post-transplant setting. But it's something that we're looking into investigating.
Great, thank you. There is another question online. Catherine has asked, I'm always curious after Greg's sabbatical, what new lines of inquiry or questions have come up that the Alfred wants to pursue to ultimately improve outcomes for transplant patients? Sort of what are some of the most urgent questions we want to address? And are there other learnings from work in kidney and liver that can help lung transplant? That's 3 questions, actually.
So I think the hottest topic out there, I think, is trying to work out what to do with the machines that you can put organs on to sort of look at them before a transplant. So out of the donor onto a machine, you play with it, and then you change it possibly, or you assess it and use that information to put the organ into somebody else. And I think there's a lot of variables there, from, I mentioned the temperature, right through to Nicole's work on cellular therapies, where you can say, okay, but now that I know that lung's going into that individual, if Steve can tell us what we exactly need to match or avoid a mismatch, and then can we manipulate the cells even in the organ before it gets into somebody else? And it sounds sort of hypothetical, but the reality is that the technology now exists to do all of those things, and and I think that's perhaps where the future is, changing things at the front end, so that you're not just well matched. You're actually you've actually got a superior organ, and it's actually primed specifically for you. So I do see that as being something that, ironically, lungs are very you can get right into the whole lung. There's sort of little sponge, then you can lots of arteries and things. Whereas to get to infiltrate a liver or a kidney is harder to do, to actually change the whole organ. So we may be finding more about this in the next few years than the other more common organs.
I suppose that's the thing. Learnings from the kidney and liver work, obviously, that they've done a lot of perfusion work already and are perfusing organs already, but they're not sort of changing; they're just perfusing it. But they're smaller than the lungs, and yet it's very expensive technology. How are we going to afford all of this technology that is required to be able to do this? And also the space, like you had showed that photo of that amazing operating theatre sort of set up that was that at Papworth? And
that was at Addenbrooke's.
And we have no space like that here at the Alfred. So, it requires quite a lot of investment, obviously, from a hospital point of view and resource.
But as you know, we can also we can take the starting work of some others and apply that, and we've done that very successfully in the past. So we collaborate with New York, and we're collaborating with the people in Papworth and stuff. And if they get half a start on it, we can really focus on it. We don't have to invent everything. We just have to translate it.
I don't think there's any other questions, unless anyone's
got a couple more there. I can see there's Jane has asked about bones and skin cancers.
Thank you.
Our own team here at the Alfred's looking at has just put together something on the Catherine Hackman and team on the bone density studies that people get done to prevent osteoporosis. And they've found that there's a better way of predicting the change in bone density, which in turn predicts the risk of fractures. So that's going to lead to questions as to how we better protect people's bones. So that's definitely an active area with cleverer drugs and closer monitoring. And that might even influence physiotherapy outcomes and stuff if you're clever. And the second one there, the skin cancers. The skin cancers are an absolute pain in the neck, pardon the pun, for everyone, and we're desperate to come up with a solution. And we were currently talking to the dermatology people about the people who've had. There's some belief that skin cancers might be some sort of form of infection, and so we're looking at seeing whether the Gardasil vaccine, which has been given to not everyone, but a lot of people, might have actually influenced their skin cancer rate and improved things. So that's a current project. Some patients are being treated with sirolimus on the skin, which is another immunosuppressant and could be used with transplant. So we're definitely keen for skin ideas and hunting around with our clever colleagues to do something novel in that.
there's another question from Helen Bergman. Is the current T-cell research being done in conjunction with the work that Dr. Sullivan presented in October 2020? Oh, that's pretty impressive to go back 5 years. Is it separate? So, Nicole,
I don't know what Lucy's project.
I can't remember.
2020, but it's a separate project. Yeah.
Great.
There's I may be referring to some of the work on different cell types around CMV and other type of cells that the question was, can you give the cells back? NK cells, can they be put back into a person and used as a therapy? And I think that the concepts that around putting them in with machines or doing them at a bronchoscopy and fitting that would fit with Nicole's thing. Can you influence some of these good populations, increase the good guys and get rid of the bad guys by one means or other? So I think they're sort of overlapping ideas of cell therapy and localized treatments.
Okay, I might hand back over to you, Wendy. Mm-hmm. Yes, thanks, Greg.
That's great. Always learn so much when I hear these presentations, and thank you for you all trying to put in lay terms as well for all of us who aren't necessarily medically minded. So it's great to see. So for those of you who want to spend a bit more time watching some of this back, as I said, we were going to record it, so you can stop and start at the recordings and get your head around some of the science if you want to. I just wanted to mention a couple of things, and then I'm just going to hand over to Gordon as well. So a couple of things which we're very excited about. We have our new impact report. So that's a picture of it there. I actually dropped off a bunch of these at the Alfred yesterday. So if you're in at the Alfred, we're going to try and get those out to people, but we'll have them at our events. And we also have this available online, effective now. So if you go to our website, you'll see that there's a link on our homepage to our latest impact report. So have a read. It actually goes into some of the research in a bit more detail, and you can also on our website learn a bit more about the research projects too. That's all now been updated as well for last year and this year. I want to congratulate again the recipients of the grants for this year. So these are the 5 people that received them. Julia actually got 2 of the grants. So again, a lot of that information is in the impact report and also on our website. But that's 6 projects is the most I think we've funded in a year. So we're very proud of that and congratulate all of them again. We also want to congratulate Steven Hiho, whose project in pharmacy has been selected as the one for the Jeff Gittis Fellowship this year, and that's really exciting for the Gittis family and all of us, and Steve, of course. So you'll hear more about his work and the other 5 in our next research presentation next year, because we've got to give them a bit of time to work on these things before we present. So congratulations to all 6 of those. I'm now going to hand over to Gordon. I'll just stop my share for a sec because I think I have to bring him online.
So I just want to say a big thank you to everyone that's online today, to the researchers, to everyone. I think it's important to understand that whether you're contributing financially, giving your time, sharing your expertise, or simply helping to spread the work of Lungitude, every bit of support adds up, and it's that collective impact that makes us do what we can do today possible. And with that, I've got the really easy task today, because I've got a big fat check, which hopefully will go across for $217,000, and that research that will be spread across the Alfred, the team of the Alfred, Monash, and St. Vincent's as well. So as Wendy mentioned, these you'll find out more about the research reports and the research that we're funding on the website. I've got a little bit of a problem with this check because my envelope's only that big, so somehow we'll work out how to get the money through to you. But I think this will be one of our largest contributions, annual contributions. And for those that want to know, to date, since Lungitude was founded, we've donated $1.7 million to research. So I think it's a massive effort from everyone, really, everyone involved in it. Someone's put through that they can't see Gordon, and that's okay. You're not missing out on anything if you can't see me. If you've seen everything else, that's more important. So that's all I really want to say. But Wendy did say at the beginning that we've got some really exciting news. So I'm going to pass over to Glenn.
Thanks to Lungitude for allowing us just to share some kind of hot off the press news. By way of background, you'd have heard today that chronic rejection, or what we call CLAD, chronic lung allograft dysfunction, is really the Achilles heel of lung transplantation. And we've known for a long time that when we diagnose it, it's almost too late in the process, and it's very difficult to kind of reverse it. We've known that our patients have a lot of bronchoscopies early post-transplant, and we came up with this idea a number of years ago that maybe we could look at these lung samples in the first year and see if we can identify a signature that predicts those patients who are going to get chronic rejection later on. We went to Lungitude a number of years ago with this idea, and we're incredibly grateful when they provided us some seed funding to kind of create some proof of concept around this idea. This subsequently turned into a PhD that Julia Iacona did and completed earlier this year. And on the back of the PhD, we took her data and put that into a submission to the Medical Research Future Fund. And we're absolutely delighted a couple of weeks ago when we heard that grant was successful, and that starting next year we'll be running a 4-year project, and we'll be receiving just under $1.8 million of funding to really unravel the causes of chronic rejection. The study team is shown there. It will be headed up by myself and Ben Marsland. Ben is a professor of immunology and runs a research lab here at the Alfred. And you can see that the success of this application, we believe, was largely funded by our study team that includes our chair of ceremonies today, Wendy Jenkins, and we're delighted that she joined us as a co-investigator on this grant, and her job will be to really ensure the point that Bron made earlier, that there is a patient-focused angle to this grant, and that whatever we find out in the lab really translates to the patient and improved outcomes in transplant. So the next 4 years, we'll be looking forward to sharing the outcomes of this great study with you. Thanks.
Thanks, Glenn. That's great. Yeah, excellent news. I'm only a tiny seed of this thing. I'm even a bit embarrassed my name's on there, but we're very excited that the RFF grant has been approved. It's going to be announced more officially in the coming weeks, so we'll then put some posts out about that as well. But this is one of the things that we love to do, is the seed funding to show proof of, so that they can then go on to do other things. Lungitude's actually been a consumer rep on a couple of project submissions this year, which really is a new sort of area for us to be in. So we're really excited that, if our foundation can help lend some gravitas to some of these submissions, and by all means, we're very happy to do that. So that's something else for anyone who's on here who's medical minded, who is looking for consumer representation. Please reach out to us and have a chat about that. With the last few minutes that we have to go, I know we've got a couple of presenters who might need to jump off, so if you need to do that, please do. We're just wrapping up with a couple of housekeeping things. So we have our next Lungitude long lunch. This is our infamous lunch that we have each year, which is a lot of fun. And this year we had it in the Botanical Gardens. Next year we're having it down at the beach in a beautiful venue that's recently been renovated. So please put that date in your diary if you'd like to join us, the 29th of March. It's a Sunday. We also have our peer support continues to grow strength by strength. We're getting people from all over Australia now interested in joining our peer support. So if you're a patient, a caregiver, a family member who's looking for support, or pre-transplant, you're very welcome to reach out and join. Every new peer group member who comes to their first meeting gets one of our lions, and all of the new patients do as well. If you're an older patient and you really love one, just send me a note. We had to sort of start at some point, but we've got a few there to share. And then finally, just another thank you to everyone again who has helped us. Big shout out to our board and committee who are behind all the things that we do. And a thank you to all of you for jumping on. I know everyone's really busy, so to give up an hour and a half of your time is very appreciated, and a big thank you to all of our presenters and Bronwyn Levy as well for hosting. Thank you again for what you do, and it makes a big difference to patients' lives and all their surrounding family and friends. If you would like to contact us about anything, our contact details are on the website as well, but please reach out. If you've got any personal lung transplant medical type questions that rose from today's presentations, please to have a chat to your specialist in your next appointments. I've just got the Alfred ones there, but I know we've got people jumping on from all over Australia. And please follow us on Facebook, LinkedIn, Instagram, and if you have any ideas or want to run your own fundraiser, then please reach out. So on that note, I will finish the official side of things, and thank you again for everyone's time.